The second major stage of protein synthesis is called translation, and translation builds proteins by translating mRNA into a sequence of amino acids, and an amino acid sequence is referred to as a polypeptide. It's not until that polypeptide then folds into a particular shape that it becomes a protein. The first process of protein synthesis is transcription, in which you take a DNA molecule and you code for an mRNA molecule, and this occurs in the nucleus. The product of that is a single strand of mRNA, or messenger RNA. The next step, which occurs through translation, is taking that mRNA molecule and translating that into a protein. The next stage of protein synthesis is called translation, and this is where you take that mRNA molecule, which was a product of transcription, and you code that into a polypeptide, which is just a sequence of amino acids, a group of individual amino acids that have been joined together in a long chain, and then eventually that folds into a protein. Now this process takes place in the cytoplasm. There are 20 different amino acids that are used to form polypeptides in eukaryotic cells, and these are coded for by a specific codon. And a codon is simply a sequence of 3 nucleotide bases. So for example, this nucleotide base sequence of AAG specifically codes for the amino acid lysine. UCA codes for serine, and GUC codes for valine. And in this way, mRNA is used to code for these individual amino acids that then bond together to form your polypeptide. In order to determine which codon codes for which specific amino acid, you use this table called the genetic code. The genetic code is read by looking at the first letter of the codon on the left-hand side of the table, then looking for the second letter of the codon on the top of the table, and finally the third letter of the codon on the right-hand side of the table in order to figure out which specific box you're looking in, and then determine which amino acid that codon codes for. So for example, let's look at AAU. So let's look for A on the left hand side of the table, and I can see it's here down in the third row. I then look across the table until I get to A on the top of the table. So now I know I'm looking at the third row, but also at the third column. So now I'm looking in this box here. Now there's still 2 amino acids in here, so in order to figure out which one AAU codes for, I then have to look for the very third letter, U, on the right hand side of the table here. And that's going to direct me to this code here, AAU. So I now know that AAU codes for asparagine. When you look at the table carefully, you'll notice that there's more than one codon that can code for a specific amino acid. So for example, proline here can be coded for by 4 different codons. Other amino acids like glutamine might only be coded by 2 different codons, and tryptophan is actually only coded for by 1 codon. The table also includes the start codon, which is AUG, and the 3 different stop codons, UAA, UAG, and UGA. And this start and stop codons, which determine where coding starts and stops in both transcription and translation. Translation requires 3 key things. It requires mRNA, tRNA, and ribosomes. mRNA is the product of transcription that encodes the proteins, and mRNA contains those 3 sequences of bases referred to as codons that are code for different amino acids. tRNA is a molecule that binds to the mRNA codon and to an amino acid at the other end. So this image here shows a tRNA molecule, and at one end of that tRNA molecule is the amino acid. At the other end of the tRNA molecule is a 3-base sequence referred to as an anticodon. And you can probably guess that the anticodon is the part of the tRNA molecule that's going to bind to the codon in the mRNA molecule during translation. The other important component needed for translation are ribosomes. And ribosomes are built of both RNA and proteins. And they can be found free in the cytoplasm or attached to the rough endoplasmic reticulum from which it gets its name. Ribosomes are made of 2 subunits referred to as the small subunit and the large subunit, which are also referred to as the 40S and the 60S subunits. Translation occurs in 3 steps, the first of which is called initiation. And in initiation, the leader sequence, which is at the 5' end of the mRNA molecule, bonds with a small ribosomal subunit. Next, an initiator tRNA with the anticodon UAC pairs with the mRNA start codon of AUG. Finally, the large ribosomal subunit attaches to the small subunit to complete the ribosome and complete initiation. The second step is called elongation, and this is when a tRNA molecule carrying the second amino acid then binds to the second codon. So this particular amino acid, which has the anticodon CCU, binds with the codon GGA. Then these 2 amino acids bind together, starting to elongate the polypeptide. Elongation continues when the ribosome moves down the mRNA by 1 codon in the 3' direction. That's when a third tRNA molecule enters the ribosome and aligns with the previous 2 tRNA molecules. When this tRNA molecule, this third tRNA molecule, aligns with the previous 2, it enables the amino acid that is attached to it to bond with the previous 2 amino acids, therefore elongating the polypeptide chain. And this process is helped by proteins called elongation factors. The third step in this process is termination, and this is where elongation halts at a stop codon, which can be the UGA, UAG, or UAA. In this example here, it is UAA. Proteins, called release factors, then bind to the stop codon, causing the release of the very last tRNA from the ribosome. The ribosomal subunits, the small and large subunits, then separate from each other and are recycled, and the protein is released. Protein synthesis can be extremely fast, with a cell producing hundreds of thousands of proteins per second. This can occur because multiple copies of each mRNA and dozens of ribosomes can actually simultaneously bind along the length of a single mRNA molecule, as shown in this image here. This assembly line can make many copies of a protein from the same mRNA molecule.